Now, I've been a Komen Scholar now for 3 or 4 years, and it's really been incredibly helpful. I study a kind of breast cancer called HER2-positive disease, and what we're trying to do is understand why cancers become resistant to therapy. And we've done that in a number of ways, and in some ways we get support from pharmaceutical and biotech industries because we're doing clinical trials. But when we're trying to really look at the mechanism of how these cancers become resistant to therapy, that's a harder thing to get funded. And I have been very fortunate to get funding from Komen to support that kind of work, which, again, is something that's a little bit harder to write grants for, especially when we initially didn't have strong data to support a project, but the Komen seed funding for this really has paid off, and we've just now are starting to get data from patients who've had HER2-positive disease that's grown resistant to current therapies, and what we've done through Komen funding is do biopsies of the metastatic tumors, again, in patients who are now refractory to the kind of current therapies we have, and are now looking at the DNA of those metastatic tumors. Essentially, we're able to sequence all of the genes of each of those tumors, which seems like a very straightforward thing to do to try to understand why the cancers have become resistant, but in fact this has never been done before on any kind of large scale, and Komen really made that possible, and we recently presented some of our data at one of the recent breast cancer meetings, and we're getting more data now to really try to elucidate why these cancers become resistant. So one of the things that my group has been working on is to try to develop new drugs specifically for this type of breast cancer called HER2-positive. And HER2-positive is not a rare type of breast cancer. It's about 20% of all breast cancers. So it's a, unfortunately, relatively common problem. And while we already have some drugs that work on this type of breast cancer, eventually patients develop resistance. And so we've been trying to develop new approaches when patients become resistant to the current therapies. And a lot of the work has been on this drug called TDM1. And TDM1 is a new kind of drug. What TDM1 does is it takes this very specific antibody and links it to a very potent chemotherapy. A chemotherapy that would be way too strong to give just as a regular drug because it would cause too many side effects. But because the very potent chemotherapy is linked to the antibody takes it directly to these HER2-positive cancer cells. So you are able to deliver a lot of this very strong chemotherapy directly to the cancer cell, but because the chemotherapy is attached to the antibody, it doesn't damage normal tissues. The drug was very effective in HER2-positive cancers, even those that had become resistant to all the previous therapies. But at the same time, it didn't cause the kind of normal chemotherapy side effects that happen when you give chemotherapy by itself. So patients with TDM1, they don't lose their hair, they don't get very tired, they don't get nauseous, they don't become susceptible to infections. And it's all because this drug is very specific for the HER2-positive cancer cell. And so through a lot of different trials, starting again in 2006, we were able to show that TDM1 was more effective than standard therapies and with fewer side effects, and that led to the approval of TDM1 in 2013. And since then, we've been trying to further work on TDM1 in various trials, looking to see whether we can combine it with other drugs to make it work better. We really want to leverage this ability of TDM1 to be so specific to the cancer cells. And so what we're doing is right now we have a randomized trial that myself and one of my colleagues, Sarah Talani, is working on, which is looking at 500 patients with relatively small HER2-positive cancers, and they're being randomized to chemotherapy and trastuzumab or Herceptin, which is one standard, versus just TDM1 alone for 1 year. It's going to take a few years to get the results, but again, we think it's a great way to take advantage of TDM1's properties